Okay, I think we're ready to begin, right?
Yeah, just a minute. It's processing now.
Okay.
Yeah, we are live, good to go.
Okay. So hi, everyone. Welcome to the last and closing session of Scimplify 2021. I'm Shriya Palchodhary, a graduate student at TPFL, Switzerland, working on neuroscience. And it's been an absolute pleasure to have you all join us for these 2 days packed full of events for young science enthusiasts. We really, really appreciate your continued support. And so we began Scimplify with one small thought, and that gets reflected in what Professor Eve Marder once said in an autobiographical piece. And she said, sometimes when I walk back to the rig rooms and watch recordings of the rhythms of our preparations, I find myself in the same state of wonder that I first felt as a beginning graduate student. Peeking into the mysteries of life never gets old. And so who better to get us excited about science the way she still feels about it today, after a long and spectacular academic career in neuroscience. So we at Biologically Speaking are superbly excited to have with us today Professor Eve Marder, Victor and Gwendolyn Beanfield Professor of Neuroscience from Brandeis University, USA, who is not only one of the most widely known and revered neuroscientists of our time, but also a champion of inclusivity, diversity, equity in science. Professor Marder received her BA from Brandeis University in '69 and subsequently completed her PhD studies at University of California, San Diego. She has spent her career unraveling the role of central pattern generators, neurons that generate movement of varying kinds, and the role of neuromodulatory effects on them. Her model organism of choice is interestingly the lobster and the crab, and probably she will tell us more about it today. She completed her postdoctoral training at the University of Oregon in Eugene and the École Normale Supérieure in Paris, France. Professor Marder has been at Brandeis since '78 as a faculty member in the Department of Biology. Through her distinguished career, Professor Marder has garnered many honors, some of which are the Society of Neurosciences Ralph Gerard Prize, as well as the Women in Neuroscience Prize, the Lifetime Achievement Award, Merit Award from the National Institute of Mental Health and the National Institute of Neurological Disorders and Stroke, and so on. She's also been the president of the Society for Neuroscience and been an editor at eLife. The list of her accolades and achievements are endless, so I will stop here because we'd rather hear what she has to say. And today, she will talk about something very interesting, the journey from basic neuroscience to climate change. A very, very warm welcome to you, Professor Marder, and the stage is all yours.
Oh, thank you very much. And can you all hear me?
Yes, we can.
Yes, okay. So, yes, I want to tell you how it was that we've come to worrying about the temperature of the ocean right outside of my window because of the effects it has on the animals we study and what those implications are for all life, all neuroscience. But there are some really, really fundamental insights into basic science that we can take by looking at the world around us. So first, before I start, I would like to somehow or other get sometimes in the beginning doesn't want to advance there. This was my lab a few months ago. Some of these people have left and some of them, and we have some new people, but you'll notice all the different kinds of faces. And this includes Sonal Kedya, who actually is Indian, and a number of other people of Indian American descent. I'm extremely lucky and fortunate to have had a very large number of talented people work with us. And my present lab is shown here on the upper left. And then there are people who are now in graduate school and other positions. A large number of people have gone on to industry and governmental positions. And then also a very large number of people have gone on to run their own labs, including down here, Václav Thermulai, who's at NCBS. And she taught me a great deal about science and about life as I worked with her over the years. And then I have some long-term collaborators. So the problems I want to be talking with to you today include how does a circuit, or how, whoops, I don't know why things, how does a circuit, I have to get rid of some stuff, it's getting in my way. How does the circuit's performance depend on the properties of the intrinsic properties of the cells and their synaptic connections, and why can't I get rid of this? And then how tightly tuned do the parameters that govern synaptic strength and intrinsic properties need to be for good enough circuit behavior, or how different are the underlying structures of normal healthy brains? These are questions that all of us care about. And then how is individual variability related to resilience to challenge? And this is really the question that we're most studying today. Okay, so as you heard before, something really bizarre. We've been using the crustacean stomatogastric nervous system. This is a preparation that comes from lobsters and crabs. And I started working with this when I started graduate school, and I've been working with the system since, because it has certain very important and useful attributes, including, and most importantly, as you'll see in a moment, all of the cells are individually identifiable. That made it possible to come up with a connectivity diagram. There's small numbers of neurons, which means it's possible to record from many of them at the same time, in our case, all of them. And it produces, this is an example of a central pattern generator circuit, so it produces a very easily recordable and characterizable motor output. Again, and these features made it really possible to do all kinds of work that just wasn't possible for a very long time with the vertebrate neurons or vertebrate systems. So here's the crab. This is Cancer borealis. These animals live in the North Atlantic, right outside my window. I actually live on the waterfront. And so these animals live in the North Atlantic, very close to Boston, where I live. The animal you're seeing is an adult. It has a carapace of 3 or 4 inches across, and it's been alive for a number of years, and therefore it's seen many cycles of water temperature, and this will become very important as we go along. The stomach of a crab is right there up here in the yellow circle, and what the stomatogastric ganglion produces the rhythms that move the muscles of the animal's stomach. But right now it's hidden underneath the carapace, which makes it inconvenient. So what we do is we remove the stomach from the animal, and this is a side view of the stomach. Up here you see the stomatogastric ganglion. In crabs it has 26 or 27 neurons. Most of them are motor neurons that send their axons out nerves to innervate 40 pairs of striated muscles of the stomach. And those stomach muscles and their movements are what the rhythms are that you'll be seeing in a minute. Additionally, the stomatogastric ganglion receives modulatory input from a number of descending pathways, and you'll see those in a second. So after we take the stomach out of the animal, we dissect the nervous system off the surface of the stomach and pin it into a Silgard-lined petri dish that's now superfused with physiological saline. And in this configuration, you can see the stomatogastric ganglion right in the center and these motor nerves, and you can see all these branches. And by putting extracellular electrodes on each of these individual branches, we can identify the neurons that used to go in this branch, or that go in this branch, that used to go to this muscle, or this muscle, or this muscle, etc. And it's the basis of those projection patterns that we identify the cells that we record. And so this shows you exactly to how we continue. So we use sharp microelectrodes to record from the somata. And here we're showing 3 sharps, but it's routine for us to put 4 and sometimes even more sharps into neurons in the stomatogastric ganglion. And using those intracellular recordings, we see membrane potential as a function of time. in these simultaneous recordings. And also at the same time, we record extracellularly from the motor nerves, and it's the recordings of the extracellular action potentials on these motor nerves that allow us to identify the cells when we record from them intracellularly. So here we see an example of the pyloric rhythm. The pyloric rhythm is a triphasic motor pattern, and it has a sequence of LP, PY, PD, LP, PY, PD. Now, before going any further, I should tell you that the pyloric rhythm is continuous at all times in the animal. So it's ongoing the same way your respiratory rhythm is ongoing. And this is one of the reasons also why it's been so incredibly fruitful, because we can look at it and know what its frequency is. We can use to measure the number of spikes per burst or the phase relationships, so the patterns that it's producing, and know when it's actually behaving normally or somewhat normally, or when it gets disrupted or disturbed by things we do. So the first thing that people did when they started working on the pyloric rhythm was to try and establish a connectivity diagram. That's what we called it in the early days, connectivity or wiring diagrams. Today we call this a connectome. And this is a simplified version of the pyloric rhythm. There's one neuron called the AB neuron. It's an interneuron. And it's an intrinsically oscillatory neuron. It's strongly electric coupled to the two PD neurons, these dilator neurons. Notice there are two of those. And then the AB and the PD together rhythmically inhibit other neurons in the network. So it's important for you to remember that LP and PY fire and rebound from inhibition. And rebound from inhibition then is the important timing cue in the generation of the pyloric rhythm. So the way this works in a simplified fashion is this pacemaker kernel oscillates, and you can see that here. These two cells rhythmically inhibit the LP and the PY, ensuring that they fire out of phase, and then LP recovers from inhibition, keeps shutting off PY, but also inhibits the PD, and eventually PY recovers from its inhibition, shuts off the LP, and the whole cycle repeats itself. Now, I'd just like to make one comment at this point, and to tell you that if I had shown you this connectivity diagram and not shown you the dynamics, there's absolutely no way you could have predicted from this connectivity diagram what this network was doing. And that's because what's missing in this connectome, or any connectome, are the details of which voltage and time-dependent currents are found in the cells, what the strength and time course of the synapses are. And without all that detailed biophysical information, you'd be hard-pressed to go from a wiring diagram to dynamics. And so a lot of what we've been doing in the last number of years is working on all those biophysical mechanisms to try and understand how this wiring diagram supports these dynamics, and how neuromodulators that change some of these properties, how they also change the properties of the network that ensues. But before going any further, I would just like to show you as a lead-in or segue to the most important messages of my talk. I'm just going to show you a very simple demonstration. Anybody who's built computational models already knows that there are many ways you can get the same output pattern with very different underlying components. So here we see PDL-PPY. These are models that Leandro Alonso made just for the purpose of making this point. And so one set of parameters gives you a perfectly nice pyloric rhythm. Here's another model with different sets of parameters, and you have a perfectly nice pyloric rhythm. Here's another model with another set of parameters. Here's another set. And then what I asked Leandro to do was to take each of these four models and just block all the synapses so we could see what the intrinsic properties of the cells were in the absence of their synaptic connections. And notice in this case, in A, all three cells were firing tonically. In B, the PD cell was firing in bursts, and the other two were firing tonically. In C, the LP was firing in bursts, and PD and PY were firing tonically. And in D, two of these cells were firing in bursts, and one of them was firing tonically. So that immediately tells you that there are many ways to produce a similar rhythm, and we call these degenerate patterns. Now, it turns out that biological experiments in the lobster and crab show that the lobster and crab actually uses this solution, with the AB and the PD cell being oscillators, or can be oscillators in isolation, and LP and PY usually fire tonically when they're isolated. But that doesn't mean that a different animal, a different species, a different part of the brain couldn't use a different strategy. I suspect that this is probably one of the more stable strategies, and that's why the animals actually use it. But I'd like to just continue to this notion of building multiple models, because everything I'm going to show you today builds on work that Astrid Prince started in a paper that was published in 2004. She was a postdoc in my lab at the time. And what she wanted to do is she wanted to build good models for the pyloric rhythm. And so she started out with models that we had in the lab, and she started fine-tuning, hand-tuning them, and she was going crazy. So instead, she generated a family of 1.7 million model neurons, in which each model had 8 conductances, and she varied each of those conductances. She chose 6 values for each of the 8, and she simulated all against all, and then she ran all those models and then classified them. And from the behavior of those models, she chose 5 that she thought were good pacemaker kernel models, 5 that she thought were good LP models, and 6 that were good PY models. And she included in her criteria that these models should be bursting and these should be firing tonically. Then she chose values for either 5 or 6 levels of the conductances for all of the synaptic connections in the ganglion. She simulated all against all. So she created a population of 20 million model networks, and then she ran them all, and then she sorted them on the basis of their behavior. So if that circle represents her 20 million models, 19% of them were what she called pyloric-like. That is to say, they were triphasic in the right order, and that's important. And then she found that 2.4% of them she called good pyloric models. And what she meant by this, and this is a really important point, she compared them to data that Dirk Bucher had recorded from 99 preparations of lobsters. So these are lobster data. And what Dirk measured was the burst duration, the burst period, the phase relationships of all the cells, the number of spikes per burst, et cetera. And then he plotted all of those things from the 99 animals. And so what Astrid was looking for were all the models that would fit into the distributions that Dirk had recorded. So she was not looking for one model that would represent the mean of all this biological data. Instead, she was looking for all the models that would fit into the same distribution. And that's where the 2.4% of pyloric came from. Now, I think it's very, very important because this tells you, this was actually at the time a paradigm shift, because she really said that the right model, quote unquote, for the pyloric rhythm is not a single model, but multiple models. And because it's the multiple models that properly represent the distribution in the biological systems. So what can we learn from this? So here are 2 of her models. Here's model circuit 1, model circuit 2. But now, because these are models, one can look at the underlying parameters. And what you see here is one of the calcium currents in the ABPD cell model is big in model network 1, small in model network 2. The PYLP synapse is small in model network 1, big in model network 2. The H conductance is small in model 2 and big in model 1. So you can see this set of parameters and this set of parameters are very, very, very different, but nonetheless, both are capable of producing very similar behaviors. So this immediately said, now in a much more detailed fashion, that one might imagine a great deal of variability in each of the underlying conductances, but nonetheless, there can be subsets of these that can work together to produce a very similar or degenerate set of pathways. And so we call these degenerate solutions because they have different underlying structures. Now, the minute you start thinking about this, you realize that it's both very gratifying as a biologist to see this kind of functional degeneracy, because that tells you that the biological systems don't have to fine-tune everything to a very, very, very high precision. But you also start realizing that to the extent to which these solutions are very different, that means that there will be in the universe of all possible perturbations that will separate them. So then the question is, if I take this seriously, and I said to myself, if crabs had this same kind of variability in their underlying structures, will that make them susceptible to the perturbations that they have to deal with in their everyday life? And so that's what basically set us out on the path we're on now. So the questions we started working on were, how variable are the parameters in real biological networks? Do perturbations reveal cryptic animal-to-animal differences? That would be differences that you wouldn't see by just looking at the output. And then how reliably can animals with intrinsic variability respond to perturbations? So the first piece of the puzzle came, and we had actually had some data from before this, but it was very nicely shown in this paper. Dave Schultz and Jean-Marc Ayer were two postdocs in the lab. Jean-Marc recorded from two animals. There's one LP in each animal. So this is the LP in animal 1, the LP in animal 2. And when he overlays them, you can see how similar they are to each other. But when Jean-Marc went into 2-electrode voltage clamp, and now he's recording the calcium activated potassium current in the blue animal, and you can see it's much bigger than the one in the green animal. Likewise, the A current, the transient outward current, is bigger in the blue animal than in the green animal. And in the data set that they collected at that point, you can see there's about a 2 to 4, 2 to 5-fold variation in the conductance densities measured in voltage clamp for these three potassium currents. Then after Jean-Marc did the voltage clamp experiments, Dave Schultz individually dissected the neurons, and he used single cell PCR to actually characterize the mRNA copy numbers in those same cells. And you can see here, BKKCA, that has, again, that range of, here it looks like it's a little more than 4-fold range. And SHAL, that's one of the genes that's important for IA, and SHAB. And when Dave and Jean-Marc plotted the conductance, which is the protein, as a function of the mRNA, you can see very strong correlations. So in these animals, for at least some of the genes, there's a very strong correlation between the mRNA copy number and the conductance. So that was early days. These now are data from a more recent study using RNA-seq. That Dave Schultz's lab and our lab collaborated on. And now we see the mRNA distributions for many more channel genes, now in PD cells, but basically the answer is the same no matter what cell type you look like. And again, you see this variability, which is this 2 to 4, 2 to 5-fold range in the same cell type across animals. At the same time, and this is the paradox, this is what I want you to hold in your mind. If you record from the animals that would have gone into this variable channel number, you see the motor patterns are remarkably constant. These happen to be 4 animals that Maru recorded. She just picked these recordings from 4 random animals. And you can see that the pyloric rhythm is remarkably robust, even though every cell in the pyloric network would show this kind of distribution or variable distribution of channel genes. Okay, so that is the basic puzzle. You have these variable underpinnings, and yet the behavior itself is very robust. So just summarizing that, individual synaptic and intrinsic parameters can vary 2 to 6-fold across animals, even when their networks have similar behavior. And this makes the prediction that there may be hidden or cryptic differences that may only be revealed by extreme perturbations. So we started thinking about what kinds of perturbations would we be able to look at that might actually help us understand this problem. So I'm trying to figure out what time I started, but I can't. But anyway, to continue. So the perturbation that I'm going to be talking about to you today is temperature, because temperature is a perturbation that influences the properties of every single membrane protein, every single protein in the cell, actually, not only membrane proteins. And we can easily characterize it with a Q10, which is shown here in this exponential. A Q10 of 1 is something that's a process that's completely temperature independent. A Q10 of 2, and I was taught when I was a kid that biological systems all had biological processes at Q10s of 2, and that is actually true for some things, but not for everything. A Q10 of 3 actually grows much faster as you increase the temperature. Now, just to put things in context, ion channel genes and ion channel proteins have Q10s that range from about 1.5 to about 4. And now we're talking about the channels that are going to be important in the dynamics we're talking about. And that includes the conductance through the channel, as well as the activation and inactivation rates that govern the opening and closing of the channels in response to voltage and time. But if you start thinking about it and you have a neuron that's, let's say, bursting, you immediately realize that as you start changing temperature, if the behavior of the cell depends on the coordinated opening and closing of many channels, you might imagine that things would go awry or start messing up as you start changing the temperature if the processes all had different Q10s. And that's exactly what happens if you start building models and assigning Q10s randomly. And that can be seen, maybe. I don't know why. This is a picture from Lamontagne's 2010 paper that Jonathan Kaplan started building models of bursting neurons just to show you the problem. This particular model at 7, 11, 15, 19, and 23, it maintains its bursting behavior over that wide range of temperatures. However, so this was one set of parameters. However, models with the same kinds of currents, but with different Q10s assigned to different processes, did all sorts of other things, including not maintaining function over that temperature range. And you can see as you go through these that they show all different kinds of diverse behaviors. So now what happens when we look in the biological system? And these are data that Lamontagne collected in our first temperature paper. So he's recording the Pyloric Rhythm with extracellular. Here you see PD, LP, PY at 7 degrees. And then you change the temperature to 19 degrees. You can see PD, LP, PY. And you can see the rhythm is maintained. It's gotten faster as you'd expect. And here he just plots this is a semi-log plot of the network frequency as a function of temperature. It has an apparent Q10 or an emerging Q10 of about 2.3. But the most extraordinary thing that Lamont's original data showed us, and here what he's doing is he's plotting the phase relationships of, here's the time that the PD, if you start with the PD on is 0, when the PD turns off, when the LP turns on, when the LP turns off, the PY turns on, the PY turns off, you can see that the phase relationships are remarkably constant over this entire range of temperature. And this is quite remarkable because the phase relationships, that is, say, the relative timing, will depend on the operation of many different kinds of conductances. But if you think about what the animal needs to be able to do, what you would need to be able to do is you need to be able to maintain the appropriate phase relationships of a motor pattern regardless or independent frequency. And so that's what these animals can do. And that's just shown a little bit more dramatically in this next slide, which shows now intracellular recordings that Lamont made at 7, 11, 15, 19, and 23. And then Lamont did an overlay of the 7 and 11 and 19-degree traces by scaling out time. And you can see the waveforms also overlay incredibly well. So this is telling you that all the different, somehow or other, the crabs have solutions that allow the currents to maintain their function over this range of temperatures. Now, one of the ways they likely do this can be visualized now in a model. And I just want to show you this. This is a model that Leandro Alonso made using a method that he developed for visualizing the contribution of different currents to a cell's dynamics. So here he's showing you just the last spike in a burst. This is a bursting neuron. He's just showing you the last spike at 10 degrees and at 25 degrees and every temperature along the way. And in this way of visualizing, he's showing you the total outward current here in black, the total inward current down here in black. And then up here, you're seeing the proportional contribution of each of the outward currents in different colors. And here, the proportional representation of the inward currents down here. I just want you to look very quickly at what's happening at 10 degrees. The purple is the A current, that's the transient outward current, and it's big at 10 degrees. And as you go higher and higher in temperature, notice that the A current's contribution is decreasing. If you look at the calcium activated K, which is the orange current, you can see it's relatively small or modest at 10 degrees. And as you go up in temperature, it's getting larger and larger and larger and larger. So this is a model that can maintain perfectly good bursting behavior over a wide range of temperatures, but it does that by changing the relative contributions of different currents to those processes, and it does so smoothly. So our hypothesis is that one of the reasons why you have so many different voltage and time-dependent currents with overlapping functions is precisely so that biological systems can make smooth transitions through mechanism such as this. And you see that again down here with the undercurrents. So I'd like you to keep that in mind. This doesn't require that all the channels have the same Q10s, but that they have functions that they can transition across. Okay, so how much time do I have? Where?
You can take your time. It's okay.
I'm okay? Okay, I want to show you a couple of recent, oh no, maybe I'll skip these anyway. It's sort of a complicated story. Okay, what the next thing that I want to go back to what Lamont noticed is that when he went in his original study, is when he went to temperatures higher than about 25 degrees, he started seeing what he called crashes. And here are 4 animals, recordings from 4 animals. Animal 1, 2, 3, 4. And notice that each of the animals is showing a crash, but it's showing a crash with different dynamics. Now, this is exactly what we would have predicted if each animal had a different set of underlying parameters for its synaptic and voltage-gated channels. You might expect that when the network broke down, that it would break down differently. So Leandro Alonso used genetic algorithms to create families of models that would be temperature robust between 10 and 25 degrees, and then break down at 30 degrees. And so this just happens to be the equations that describe what he did. But you can see this particular model at 10, at 15, at 20, and at 25. The model is working beautifully. It's doing just what Lamont's data did, which is it goes faster as you increase in temperature, but the phase relationship is made constant. And then at 30, it crashed. Now, Leandro created a number of these models, and then he just selected from that some random 4 of them, just to show you that the models now also, because they have different underlying sets of conductances, they also crash when they do crash with different dynamics. And you can see here model 1, model 2, model 4, model 3. They're all crashed at 30 degrees or whatever the temperature is, but they're crashed differently. And they crash differently in ways that are very reminiscent of the biological data. So that tells us, or that leads to again, reiterates the speculation that, remember, at 11 degrees in the control, all of these models and all of these behaviors would be perfectly fine, but we're only seeing the effect of the differences in conductances, et cetera, when we do an extreme perturbation. Now, when Lamont first started seeing these crashes, and later when we started trying to characterize them, it became very difficult to figure out how to visualize the crashes in a way that would give people a sense of what was going on. So Sarah Haddad wanted, and I'll show you in a minute, she wanted to study the effects of neuromodulation on robustness to temperature crashes. But she wanted a way of just sort of being able to show you what was going on. So she took rhythms of various kinds, including a normal rhythm seen here with this purple, or a very slow pyloric rhythm, as you see here in yellow, and then all sorts of disordered rhythms. And she characterized them. So here she was talking an intermittent triphasic one, she's labeled brown, just PD alone in green, atypical firing, that's just sort of garbage firing in light green, silent in black, etc. So she is going to use these sets of criteria to characterize crash rhythms and what they look like and to study them. But before that, I just want to show you one piece of, again, important data to start with. I mentioned the descending modulatory inputs that come from these anterior ganglions, and there are 25 pairs of descending modulatory neurons that send axons into the stomatogastric ganglion. And when those axons and those modulators are present, you see the really well-behaved pyloric rhythms that I've been showing you up to this point. So this is the triphasic pyloric rhythm, as you've been seeing. If you now, however, cut that nerve, you see the rhythm can either stop or else it goes often much more slowly. And Al Hamoud character sort of did a pretty fine statistical comparison of these. So this histogram describes the pyloric frequency of 123 animals. So you can see the frequencies vary over that threefold range. But then after he looked at the decentralized frequencies, those are the red ones, you can see the whole histogram has moved to the left. There's still a few outliers that are going pretty fast, and some of them here are stopped. And the other thing that happens is the characteristic phase relationships break down. So what Sarah said, she said here, Lamont studied the effects of temperature on the normal rhythm with modulators intact. We see here that the modulators, in the absence, the rhythm becomes slower and also much more variable. So Sarah said, well, what if I study the effects of temperature on the decentralized rhythm, and then I apply back some of the modulators that were removed? And so that's what she did. So here I'm showing you 3 of Sarah's preparations, animal 1, animal 2, animal 3, from 11 to 31 degrees. Animal 1 had a perfectly good pyloric rhythm. It was one of the good ones when it was decentralized. And you can see as you go up in temperature, it maintained its activity. Animal 2 started out with a pretty good rhythm, but it became disordered to some degree at high temperatures. Animal 3 had a totally disorganized rhythm to begin with. That was one of the ones on the left of that histogram. And you can see it did not get better as temperature was increased. And this is now what you see in Sarah's color classifier. Slow rhythms, animal 1 had a slow rhythm, which then became fast, but it was good pyloric rhythm in both cases. Animal 2 started out pretty well, but then started producing all kinds of transitions into less good rhythms. And animal 3 started out not great to begin with, and then started making transitions, although notice at high temperatures, it produced a little bit of normal pyloric rhythm, and then also had many transitions. Okay, so now, Sarah then chose to study 3 modulators. I just should summarize here and just say removal of the intrinsic descending modulators reveals substantial animal-to-animal variability, and you can see that both in the raw data and in the responses to temperature. Okay, so Sarah's first modulator, it's not really this is oxytremorin, which is a muscarinic-cholinergic agonist. So the animal 4 and animal 5, you can see in the presence of oxytremorin at 11 degrees, they're both producing a normal pyloric rhythm. And as you go up in temperature, they're doing just fine. Animal 6 and animal 7, she studied the effects of proptolin. This is a peptide that's found in all arthropods. And in both cases, in the presence of proptolin, she started out with good rhythms, although in this one, obviously, it's much slower than it was in this one. And as you go up in temperature, those good rhythms persist. And that's seen here in the color plots. And you can see these rhythms are very stable in both of those modulators. And they're much more stable than they were before the modulators were produced, because remember, these guys also, in the absence of modulator, looked like the ones I showed you before. Then the next modulator she studied was serotonin. Now, I should tell you that these 2 modulators, oxytremorin and proptolin, both activate a current we call IMI, and this current tends to excite the rhythm and turn it on. So these data made complete sense. Serotonin, in contrast, activates multiple receptors, influences many different kinds of ion channels, and does so differently in every cell type. So I sort of predicted it would be a mess. And here you see Sarah's raw data, animal 8, 9, 10, 11, 12. You can see that in the presence of serotonin at 11 degrees, the baselines are quite different. This one looks pretty okay, but you can see these already look very, very different. And that's exactly what you'd expect because of the diversity of the serotonin actions. But now if you look across temperature, you can see that in no case does serotonin really help. And that's seen in the color plots, where you can see that even in the case where you start out with a good pyloric rhythm, you lose it at high temperatures. And here you start out with an okay rhythm and you lose it at high temperature. Here you start out without a good rhythm and you never get one back. Here you start out with not a good rhythm and you get it back for a little piece every now and then. Here you start out with an okay rhythm and then lose it, and then everything goes silent. So in this case, the serotonin is not compensating for the variability. Now I'm just going to end with 2 more data slides. And this is where the climate change comes in. Now, I started out showing you Lamontagne's data. And I showed you that 11 degrees, he had a nice rhythm. At 21 degrees, it was going faster. And then I showed you, not in the same exact slide, but that at 31 degrees, this preparation was crashed. Then I showed you some data of Sarah's. And I don't know if you noticed, but we went up to 31 degrees in some of hers. So in 2012, when Sarah was working in the beginning of her work, at 11 degrees, she had perfectly nice rhythm, 21, 31, and then 35. And when Sarah first saw these rhythms at 35, I was sure she had made a mistake, but actually all of her data were consistent. In that year, in 2012, the rhythms were stable up to very high temperatures. And we discovered that the data she was collecting were subsequent to a winter where the water had never gotten cold. So we put that away in our heads. Jacob Ratliff did some experiments looking at crashes in 2016. And notice here at 11 degrees, he has a perfectly nice pyloric rhythm. He has a perfectly nice pyloric rhythm at 21. But in 2016, when Jacob was doing these experiments, they were all crashing around 28, 30 degrees. And now Mara Rue and Janice Lee were doing recordings this year. And here's what they saw: 11, 21, 31, 39. So they got rhythms that were stable to much, much higher temperatures than we'd ever seen before. And what Mara did is she's gone back and looked at the crash frequencies for Jacob's data and her data. And you can see here that in 2016, 2017, the crash frequency was about 3.5. And in the more recent, it's up at 5.5. And the crash temperatures were, again, there's always a spread, but we're down closer to 30 in Jacob's data and closer to 33 or 34 in Mara's data. And that's what you see here, that all the purple dots are on top, and that most this past year, and the black dots are below. And then that's just to make Mara happy. And this is the sort of the end of this. What Mara then did is she went to the NOAA database. NOAA is the National Oceanographic and Atmospheric Agency, and they publish the surface water temperature on a number of buoys that they've placed in the ocean. And Mara went to a buoy that is in the Atlantic Ocean, about 16 miles northeast of Boston. So this is in the catchment area for our crabs. And here are the average summer temperatures from 1990 to 2020, and the average winter temperatures over the same period of time. And if you look, you can see there's obviously a trend, overall trend for warmer temperatures in the ocean. But most notably, Lamont's original experiments were shown here in green, crash temperature, and here it's green. And Mara's most recent ones, shown here in purple, up here. Sarah's, which were also high, are up here in blue, and Jacob's, which were low, are down here in black. And the same thing is true for the winter temperatures. So you can see that there's a strong correlation, and we still have to collect more data and get more data between the crash temperatures that we're seeing and the average water temperature in that year. And you can see there's fluctuations going up and down, and we're seeing that in our recordings, but the overall progression is obviously upwards. Now, the reason why I think there's a really important lesson to be taken from this is obviously in some ways it's documenting the effects of climate change and the change in the water temperature. But the most important thing that I take from this is if we had just looked at 11 degrees, we never would have known that something had changed. And that's because 11 degrees is our control temperature. We've done, thousands of experiments at 11 degrees, and we've done thousands of experiments where we've looked at lots of things, measured 11 degrees. And if we had only done our control measurements, we never would have known that there's been a really massive change in the underlying properties of the circuit that allow the animals that have been exposed to warm temperatures to acclimate in a very big way. And it's precisely because of the different underlying, the ability of this network to operate with very different sets of conductances that allows this to occur. But it also tells us that if you don't do a perturbation, you can miss the effects that are cryptic in the absence of that perturbation. So I'd just like to summarize what we've learned by looking at these crashes. Under control conditions and in response to many perturbations, all networks are well behaved. Each individual animal crashes with individual dynamics as predicted by their underlying parameter differences. Individuals with similar starting circuit performance with relatively similar circuit mechanisms in the control state can nonetheless show entirely different crash mechanisms. And this is a cautionary tale for thinking about disease, because this is, again, directly relevant to thinking about disease and what could be happening. And then differential resilience in the population is a necessary feature that comes from inherent degeneracy in multiple solutions producing similar behavior in healthy nervous systems. And then climate change may be causing changes in animals that are not easily apparent under non-stressful environments, but may only be visible under extreme conditions. Nonetheless, these cryptic changes may alter performance and produce unpredictable changes in animal behavior. And it's this last point that actually concerns me the most, because if you think about the crabs in the ocean and the humans in the California desert, who are all seeing these very unusual sets of environmental conditions, there can be changes, long-term changes that may last for quite a while that one doesn't see when you put them back into better environments. And I would stop there and thank you for your attention.
Thank you so much, Professor Marder. That was a fascinating talk. I especially liked, we talk a lot about ion channel dynamics and how it changes with temperature, but the idea that even though ion channel dynamics are so different, they end up in the neuron behaving the same way across such a varying temperature range is really, really fascinating. I had no idea. On that note, I have a small question to ask before we move to questions in the chat box. But when you say that, like when it's not, the behavior isn't changed, but the different ions, the sodium ions or the potassium channels are behaving in different ways or contributing differently to the rhythm. Is it from studies, is it from modeling, or is it from experimental data where certain ion channels or certain currents were blocked? Because is it expression of the channels, or is it the structure of the channels that are changed?
So there are three separate questions in that.
Yeah.
Okay. The example I showed you was from a model, because where you saw the change in the contributions of the A current, the calcium activated K current. And that's because it just makes the point very, very clearly. It's a little bit harder to do that technically, because to do that experimentally means you need a good voltage clamp of multiple currents over a range of temperatures. And the cells don't really like to be voltage clamped over as you start changing temperature. But the data that we have would suggest that that's exactly what's happening. That when cells can actually operate correctly, they're still doing a trade-off of, for example, which currents are doing what job. Now, in acclimation and long-term temperature treatments, there almost certainly, as part of the acclimation, are changes in gene expression. So I think one has to distinguish between the kinds of things that happen during acclimation, that is to say, the animals being out there in the ocean for 5 months at a warmer than normal temperature, that will produce changes in heat shock proteins, changes in gene expression, and everything we know would suggest that the mRNA copies and therefore also the conductances will change during that behaviors. Now, presumably, that's what we're seeing. We're finding we're sitting in the 31-degree animals in 2021. We're seeing, we're on a different place in conductance space than we were at 31 degrees in the crashed animals in 2010 or 2006. So what we're doing is we're trying to do some single cell PCR measurements and RNA-seq measurements in different years, et cetera. And Dave Schultz is actually going to go back and look at all of the PCR data in all of the cells that we've ever done as a function of year and then as a function of temperature. So we may actually be able to pull it out. We may be able to pull down to the single gene level, the same kinds of things that Mara was showing you at the end.
Oh, that's fantastic. Yeah. Cool. So there are a few questions in the chat box. The first one from Shonchari Gorai, who says, have you seen changes in pyloric rhythm in case of organisms which are desert dwelling? I think you mentioned it at the end of it. And she says, is there a fall of temperature at night? So is that a fast acclimatizing process as well?
So I don't think in the ocean that the temperature changes very much from day to night, because remember, we're looking at a very large body of water.
Yeah.
That said, the crabs we study live from temperatures of about 2 degrees to about 25 degrees normally. And they can see a 10-degree change in temperature in a given day as tides come in and out, as storms come in and out. So they're used to seeing large swings in temperature in a day, but I don't think it's day night. I think it's, a storm comes in and brings in cold water from the ocean, but they can deal with that. I think normally, they're built to deal with temperatures. They evolved to be able to deal with temperatures probably on average between about 2 and about 25 in this area. Now, if you look in the southern Atlantic, animals live there, they'll obviously be warm shifted.
Right. Thank you for that answer. So Sundaram is asking, he says, very interesting work, Professor Marder. The pyloric rhythm, what you have shown is by an ensemble method. So he's wondering what could be the picture in single cells. So I guess without synaptic connections.
In single cells, what you see, I sort of showed you some of that in the very beginning, where you see that range of, so for example, if you look at the contribution of the calcium activated potassium current in the same cell type in single cells, it can vary, call it fourfold. And that's true for every current we've ever measured. That's also true of the synaptic currents. So they vary across preparations in that same range, but the cell's activity is more reliable than the underlying conductances. So I think it always goes in the same direction. That is to say, the lower down you go, whether you go to these individual currents or the individual synaptic strengths, they vary much more than the properties of the cells do, and the network is more stable still.
Right. On that note, I have a stupid electrophysiologist's question to ask you, actually, because when we do high temperature recordings, and by high temperature, we mean like 35, 36, which is like physiological for mammals, it's in any case difficult to keep cells alive. Is it the same when you do low temperature recordings, or is it different because these are recordings from creatures which are their physiological temperature?
No, our animals are perfectly happy. Our animals are perfectly happy. Okay. Down. We can't actually get our recording systems lower than about 5.
Right. Yeah.
Our Peltiers won't go low enough.
Yeah.
No, these animals are built to be temperature robust. Now, of course, the neurons that you're recording from with difficulty at high temperature are built to be robust at 37 degrees. So the fact that they're not is a whole other thing. And it may have to do with...
it's just metabolic processes, probably.
Yeah, energy, they're chewing up energy too fast or something.
Yes. I see. So Shantari has another question. She says, are there any chances of perturbation caused by any other environmental factors other than temperature? And if any, then will climate change be playing a major disturbance for those animals, not just through temperature, but these other factors?
So we've studied several other perturbations. I just didn't have time to talk about them. We've studied the effects of pH. And obviously that's very important in the ocean as all sorts of pollutants. And we've studied the effects of high potassium. Now, with regard to pH, surprising to us, the network is very stable, very robust to pHs from 6 to 9.5. So that the pH swings that the ocean will normally see should be fine for these animals. And then we've also looked, Jacob Ratliff's data came from a paper in which he looked at the interactions between temperature and pH. because he wanted to know if the two perturbations would influence each other. And they do to a small degree, but not anywhere as much as you might expect. And actually, if you think about it, these animals probably see pH swings and temperature swings together as well. We've done a lot of work with this high potassium, which is a perturbation that, again, is global. And it's all very interesting. There are adaptations there. There's a fast adaptation there, and there's a memory effect there. But we don't yet have enough data, and there's probably an interaction with temperature as well, but we don't have that fully worked out. One would want us to do, if you wanted to be mean, to look at the effects of oxygen tension. And it's just a little more difficult to do it technically, to manipulate oxygen levels. So the three we've studied pretty carefully are temperature and pH and high potassium. And, we originally were curious to see whether you had animals that were more resilient to all perturbations, or whether there's a trade-off between resilience to temperature and there you lose resilience to pH, et cetera. But as far as we can tell, there doesn't seem to be a hard trade-off. And it doesn't seem to be that animal, it just seems to be a little mushier than that.
Right.
And it's just hard to do these experiments to really get, because you've got to keep things alive for a really long time.
Yes. Okay, Rajashi Chattopadhyay has a question. He says, do you think there are any ways to reboost damaged nerves? Yes, in the case that you showed, high temperature causes these failures, or the rhythm is lost. Is it an irreversible change, or is it not?
So, I didn't show you the crashes. I mean, when we crash them, high temperature, all we have to do to bring it back is to go back to low temperature. They come back.
Okay.
They come back. And we've done, and that's true with high potassium, it's true with pH. If you keep them for a very long time at high temperature, that's called cooking. Yeah. That's different. But no, the crash is totally reversible.
Okay. So Shaunak Shahu has a question. He asks, so how can optogenetics be useful to understand the functional circuit dynamics, particularly in systems with less completely elucidated circuits?
I guess that's sure. I mean, I don't know. I mean, we can't do optogenetics per se because we don't have a genetic organism, so we can't express optogenic proteins easily. I think optogenetics, like all techniques, are useful to the extent to which the person doing the experiments is clever. So I would say optogenetics can be useful if you're smart and do the right experiment. And I don't know what that is in whatever case you're thinking about.
No, I agree. It's a very myriad range of things you can do. And it's very difficult to pinpoint the right way to do it. So Smrita asks another question. Can I go ahead with a few more questions if you have time?
Sure.
There's a few time. Okay, thank you. So Smrita is asking, can all organisms adapt to rising oceanic temperatures, or any known thermal protective genes known to help organisms adapt to these rising temperatures?
Well, all animals, as far as I know, have heat shock proteins. And heat shock proteins react to stresses of all kinds. Interestingly, we've looked at the effects of high potassium, pH, and temperature on 3 different heat shock genes in our crabs, and they all activate heat shock genes, but they also do it differently. So there's a different pattern of heat shock genes for each of these perturbations, which may argue that there really is something different about the way in which they're reacting. So all animals have a heat shock response, which should help them deal with extreme temperatures. Some animals are going to be better. In other words, some animals are really designed to deal with a lot of temperature swings. Other animals do a lot of migration. So in that 2012 study where Sarah was getting these really robust pyloric rhythms, that spring, the people at Woods Hole complained bitterly in that summer that they couldn't find squid and dogfish because all the squid and dogfish had moved to Maine. That is to say, they had migrated. They had a strong temperature preference for cooler water. So they just left and went north. You know, I mean, it's so they're animals that actually do migrate, that have strong temperature preferences. And so animals that do have strong temperature preferences and can move to a more suitable temperature will often do so. I think the squid and the dogfish came back the next year, and the waters came back down. Yeah. So, I mean, I think in the ocean, there's probably a lot of temperature dependent migrations going on. Now, there's some things that can't migrate, you know, that are stuck where they are. And I imagine that they have a range of temperatures, and if you take them way beyond that, they'll just die. I mean,
yeah. I mean, we just had reports from the Canadian seas this year where there was so much of marine life found dead on ocean beaches, I guess, just from rising temperatures. So,
well, and also they're losing I mean, it's more complicated up in the Arctic because they're losing the Arctic shelf.
Yes.
And they're losing the places those animals live. So, I mean, it's a very complicated, and it's just terrible. It's just awful.
But thank you so much, Professor Marder, for being here. It was fantastic talking to you and looking at work that we've seen in papers our whole lives, but really interacting with you.
Well, it was a pleasure to be here, and good luck. And I hope everybody just has a wonderful next period of time.
Thank you. And thank you for sharing your Sunday morning with us.
Oh, my pleasure. Okay, bye-bye.
Bye. Okay, so with that, all good things come to an end. We thank everyone for being with us for these 2 days, and thank you for your continued support and making our event such a success, and even sharing your science enthusiasm with the contests, with all of the interactions that we've had so far through the last 2 days. I'd also like to thank the team, and please bear with me as I thank everyone who's been involved, because as you know, such an event takes a lot of effort. So from the core team, I'd like to thank Shaunak, Shorodeep, Sundaram, Sanjana, Krishnamurti, Dushyant, Rupkatha, Prabhahon, Rahi, Shoilaja, everyone for being the backbone behind this effort. From the graphics team, Debolina, Ritvi, Ucha, and Shivani for designing posters and all of our promotional content. From the editorial team, Preetha, Smrita, Shatarupa, Ankona, Revati, Rashmi, Ashwarya, Navneel for setting up all our contests and for writing up all our, especially for the Fantastic event, actually. From the operations team, Manya and Anjali, also for helping out with Eureka. From the PR team, Alberta, Nidhi, Shanchari, Sandhya, and Shailaja, who put in massive amounts of effort to get this event going. So thank you all. I hope I haven't missed anyone. I hope you've had a fantastic time and enjoyed as much as we have. And till then, see you, but hope to see you at our next webinar. But keep an eye out for us on Twitter, on Facebook, on all of our social media channels. And with that, I sign out. Thank you and have a nice Sunday.